Hello there, welcome once more to this YouTube channel. Kindly subscribe to this YouTube channel. I'm talking about molecular biology and genomics, an important subject area in medical genetics for biomedical postgraduate students, that's the M.Sc students. Of course, the PhD students and even the undergraduates will benefit from this. What are the objectives? 1. Students need to understand nucleic acid structure and interactions, signaling proteins and membrane proteins, enzyme kinetics, drug discovery, and protein design. 2. Steps in gene expression from chromatin accessibility to translation and messenger RNA turnover, including the dynamics of protein and membrane-bound organelles. 3. Candidates also need to understand cell signaling in molecular biology, the molecular basis of cancer, DNA repair, and apoptosis. 4. Students need to know protein synthesis mechanisms, especially with respect to ribosome structure, function, and accuracy translation. 5. Candidates need to know nucleosome positioning in relation to promoter architecture, promoter remodelling, also the roles of histone acetylation and the targeted acetate and acetylates, and the action of ATP-dependent chromatin remodelling machines. Right here is an illustration of DNA structure. What are the outcomes, the level of knowledge the students need to display at the end of the study of this subject area? 1. They need to appreciate that molecular biology is the basic science that has as its goal an explanation of life processes at the subcellular and molecular level. Secondly, they need to appreciate the organization of the genome, the replication, the formation of RNA, that's transcription, the processing of pre-messenger RNA, and protein synthesis, that's translation. 3. They also need to know how to relate properties of cancerous cells to mutational changes in gene function. 4. At the end, they should be able to appreciate regulation of cell form and movement, including cytoskeleton organization and generation of force and cell motility. 5. They need to know basic molecular genetic methods, including work with bacteria, PCR amplification, that's polymerase chain reaction amplification, and analysis and electrophoresis of nucleic acid. Right here is an illustration of polymerase chain reaction. What are the specific topics the students need to study and be taught? 1. Structure of nucleic acid, DNA, RNA, messenger RNA, transfer RNA, ribosomal RNA, denaturation and renaturation of DNA, the TM, that's the melting temperature, the GC content in relation to the melting temperature, that's the guanine cytosine content in relation to the melting temperature, renaturation kinetics of DNA and complexity of DNA, cut curves, satellite DNA, repetitive DNA, SNP, that's the single nucleotide polymorphism, SCR, the short tandem repeats. The second topic here is DNA replication, mechanism of DNA replication, enzymes and accessory proteins involved in DNA replication, DNA damage and repair. 3 is DNA recombination, models of homologous recombination protein machinery, homologous recombination from model organisms to humans. 4 is transcription, RNA polymerases, general and specific transcription factors, regulatory elements and mechanisms of transcription regulation, the 5' cap formation, transcription termination, 3' end processing and polyadenylation, post-transcriptional gene silencing. 5 is RNA splicing, nuclear splicing, spliceome, and some small nuclear RNAs, group 1 and group 2 introns, cis and trans splicing mechanisms, transfer RNA splicing, alternate splicing. 6 is translation, synthesis of amino acyl transfer RNA synthesis, mechanism of initiation, elongation and termination, regulation of translation, co- and post-translational modifications of proteins. 7, the last topic but certainly not the least, is regulation of gene expression, induction and repression, operon theory, the lac operon, that's the lactose operon, trip operon, that's the tryptophan operon, ara operon, that's arabinose operon, attenuation, positive and negative control, catabolite repression, regulation of post-transcription by cyclic AMP and CRP, that's the cyclic AMP receptor proteins. Check out other videos on this channel for details of all these topics. Don't forget to subscribe to this channel. Thank you for listening. Goodbye for now.